So we just discussed that the membrane is a mosaic consisting of many different pieces. Mosaic actually comes from an art term where people would make art from many different things. So for example, you can actually make a mosaic out of jelly bean. So notice each little piece right here is a jelly bean, making up this entire picture. So again, many pieces coming together to make a whole. And that's exactly what your membrane is. Many different pieces coming together to make a whole membrane. Now let's go into the functions of your plasma membrane briefly. The major function of any membrane is to be protective. So the plasma membrane is a protective barrier for your cell. It's protecting what goes in and out of the cell. It serves as receptors for communication. So like we said, on the plasma membrane are things like the glycocalyces, which serve as identity markers to help other cells communicate with this cell. And the plasma membrane, finally, is selectively permeable or has selective permeability, meaning that things have to come in and out of the membrane, but the membrane needs to be selective on what enters and exits or what transports across the membrane. For example, here, the HIV virus was able to enter the cell. Here's an example of the bilayer, and remember these proteins that are called transmembrane because they go completely through the membrane. One of the major functions of the transmembrane protein is to serve as transport for molecules that need to go into the cell. For example, these little red molecules need to come into the cell, so the membrane is selectively permeable for those little red molecules. So just to summarize what we've discussed so far with the cell, we've talked about the plasma membrane and how it has a structure consisting mainly of phospholipids and proteins, which function to protect, communicate, and transport selectively. Next, let's move on to the cytoplasm. So the cytoplasm is everything inside the plasma membrane. It includes your cytosol, organelles, and inclusions. So let's go into a little more detail about that. The cytosol. So the cytosol is basically the jelly-like fluid inside the cytoplasm. So if you were to touch it, it would really feel like jelly. And embedded in this jelly-like fluid are many proteins. Now that jelly-like fluid is a great place to keep organelles in certain positions. So organelles are the functional units within the cytoplasm. And each organelle does a specific job. For example, a mitochondria makes energy. And the third main part of the cytoplasm is inclusions. Inclusions are temporary structures mainly for storage. So they're basically sacs. And for instance, they can store things such as water or perhaps sometimes waste. But again, they're temporary and used when needed. Now let's go into a little more detail on the organelles. You've covered organelles before in your intro bio class, so we're not going to go into much detail and we're going to go through these pretty quickly. But there are 9 main types of organelles. First, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, mitochondria, cytoskeleton, centrosomes, and centrioles. Now, remember, each organelle has their own function. Now, why do we need so many different organelles, and why are they all separate? Well, think about an apartment or a house. Right? Think about why are there many different rooms? For example, in this nice apartment, we've got one bedroom, we have a living room, we have a kitchen, dining room, balcony, bathroom. Why so many rooms? Well, clearly each room serves a purpose, right? Hopefully kitchen to cook, right? Bedroom to sleep, dining area to eat, right? So like the organelles, each one has its own function. And we'll see that each organelle is isolated within its own membrane, just how each of these rooms have walls around them. Right? You wouldn't want, for instance, your kitchen mixing up with the bathroom. Right? So you want to have separation between them. Therefore, each one can serve its own function. And just like that, the organelles here will each have their own membrane to keep them separate from each other, and then they can serve their function. Now, like I said, we covered most of this in intro bio, so I'm not going to go into much detail here. But quick review. See if you can answer this. Which organelle is matched correctly with its function? A. Mitochondria synthesizes protein. B. Ribosomes make ATP. C. The nucleus controls the cell. Or D. Lysosomes package and ship protein. The correct answer is C. The nucleus controls the cell. Let's take a look and see what the other organelles do. So mitochondria doesn't synthesize protein. That's actually the role of the ribosome. Instead, the mitochondria is the powerhouse, so it actually makes the ATP. So these two are a switch. And then we'll see that the lysosomes, they don't package and ship. That's the job of the Golgi apparatus. Lysosomes actually help you eliminate the waste. So I'll go through and quickly review these organelles. So let's talk about ribosomes first. Now, you probably don't recognize what's going on here, but do you have a cell phone? Perhaps you know someone who owns an iPhone. This image I have here is actually an iPhone factory in China. So all of these workers here are making iPhones. Now, the reason why I chose this picture to describe ribosomes is this is a factory. A factory makes products. Right? In this case, the product is an iPhone. Ribosomes in your cells are factories, and the products they make are protein. So ribosomes, think of it as a factory to make protein. So their job is to synthesize or to make protein. And how they do that is based on the instructions from DNA and mRNA. Now let's look at the structure of a ribosome that helps it do it. So right here, you have the nuclear envelope, which is surrounding the nucleus. And then coming off of this in blue, you have the ER, or the endoplasmic reticulum. And then over here, you can see a real, this is an electron micrograph image of the rough ER. Now, on this ER, we say that it's rough because these little red circles that you see right here are actually the ribosomes. So the ribosomes are bound to the rough ER. That's what we mean by the structure. They are actually, ribosomes themselves are protein bound to the rough ER. Or sometimes you actually just find free-floating ribosomes in the cytoplasm. So they can be free in the cytoplasm. Next, we have the endoplasmic reticulum, and I just mentioned it, so we can see right here the rough ER. The endoplasmic reticulum is actually an interconnected membrane that's continuous with the nuclear envelope and the cell membrane. So if you notice here, here's the nuclear envelope, and then right coming off of it is the ER. And this will span all the way to the plasma membrane. Now the functions of the ER, we've got the rough, and we have the smooth. So the rough ER we know synthesizes protein. Turns out the smooth ER can synthesize also. Not protein, but they synthesize lipids. Smooth ER can also store and transport proteins and also help detox, eliminate drugs. So here's a review. Rough ER binds ribosomes and therefore synthesizes protein, and the smooth ER can synthesize lipids and help break down drugs. Now let's talk about the Golgi apparatus. So what happens to those proteins once you make them? How do you get them out? The Golgi apparatus serves somewhat like your UPS. So, not sure if you've used UPS, but hopefully you know generally what it does. Let's say you've got something you want to send. You can go to your local UPS store, and you can actually just bring, let's say I want to ship a book. I can actually just go to the UPS store with my book, give it to them, and for a nice little price, they will actually package it for me, wrap it up, protect it, put it in a box, and send it off to the right destination. Right? That's the job of UPS. The Golgi apparatus is very similar. The Golgi apparatus gets the proteins that were just made from the ER. And what it'll do is it'll package them up. So you can actually see right here, we're forming little packages, and the packages are in what we call a secretory vesicle. The vesicle is the circular package. And what it's going to do is it's going to ship it off to the appropriate location. So again, their function is to modify, so they can make changes to the protein, pack them, and deliver them in these vesicles. The structure of the Golgi apparatus is they're actually also membranes. They're flattened membrane sacs that we call cisternae, and they're actually continuous with the rough ER where we just got our proteins from. So it's all continuous together. All right, so we've packaged these proteins and we put them in vesicles and we're shipping them off. But where are we going to ship them to? We've got some options. Vesicles generally go to 3 different destinations. So now we're saying, okay, where do we ship them to? A, they can leave the cell. And how they leave the cell is through a process called exocytosis. So you can actually see this right here. Here's the vesicle, the little proteins in purple come to the edge of the cell. Here's the plasma membrane of the cell, merges in with the plasma membrane, and actually releases the proteins through exocytosis. Let's review that word exocytosis. Remember, exo, what are you thinking? Exo means exit or go out. So exocytosis is to go out of the cell. Option B. Some of these proteins actually need to go to the plasma membrane itself. Remember, we just studied the plasma membrane, and there are a lot of proteins embedded inside that plasma membrane. Peripheral proteins, integral proteins, glycoproteins. So some of those proteins that we just made over here in the ribosomes get packaged, and they come to the plasma membrane, and they stay in the plasma membrane. And then C, some of the proteins that we just made actually stay in the cell, and they'll stay inside the lysosome. So the vesicles actually become a lysosome, and the lysosomes will break things apart. So these are, for instance, maybe the proteins that we didn't need. Let's talk a little bit more about those lysosomes and peroxisomes then. So I put up here a symbol of an LAPD, the police department, and think about what the police department should do, right? They should generally go around, patrol, and make sure there's nothing bad going on. And that's basically what lysosomes and peroxisomes do. They go around, they gather up all the waste or the, quote, bad things in the cell, and they help eliminate. Lysosomes and peroxisomes, both of them have a structure. They're spherical membranes with enzymes, so they look almost like little vesicles themselves. So here's actually a real image of a lysosome, shown here in orange, and then the green would be the material that they're trying to digest or the waste we're getting rid of. The lysosome, they digest, they act like a garbage disposal. The difference between a lysosome and the peroxisome is that the peroxisomes specialize in detoxifying harmful substances. In other words, they're going to eliminate the waste that is specifically harmful. Somewhat like if we're in lab, you'll notice sometimes we'll whip out a bright red biohazard waste bag, right? What's the reason for that? It's because we're going to put things in there that we don't want in the regular waste, right? Like blood and other things. And that's, again, the goal of the peroxisome, to take care of the really potent stuff.